Hi guys, welcome to the second topic of this week, which is about transplantations of tissues and organs. So this second topic is quite brief, so we're not going to have a middle break here. Okay, it is just going to be in one part from straight to the end, and we're going to do some questions at the end like usual. Okay. Like I said, it's quite brief. Even the learning objectives are quite straightforward, basically describing some different types of transplant rejections, the roles of T cells in these type of rejections, and understand some of the basics, okay, of common immunosuppressant agents. And a lot of those immunosuppressant agents, the drugs, we're going to talk about it in other classes, okay, so in the GEM class again, so that's why I am not repeating myself in this class. So when we are talking about transplantations of tissues and organs, and particularly organs, all right, the first goal here is to have the donor's organ function normally after transplantation. That is really the main thing. Otherwise, we will not do donor organ transplant. And second, the health of the donor and as well as the recipient need to be maintained during and after the transplant. Now here we're talking about a live transplant here. Now if you are transplanting a donor organ from a diseased person, then of course the maintaining the health part is only relevant to the recipient. Okay, and also the third goal is to prevent rejections. Okay, this type of rejections or developing of an adaptive immune response, it's on both sides. Okay, we're talking about host versus graft and as well as graft versus host. Okay, here is the graph that are showing you different cases. Okay, when a kidney is being transplanted to a recipient, and what happened is the recipient's T cell would recognize it being foreign and start rejecting the transplant organ. Now, on the other hand, when bone marrow is transplanted, the B cells, the T cells from the transplanted bone marrow will start, leaking out, coming out, and attacking the recipient's tissue, so-called graft versus host disease. All right. And so here are some of the terms that need to define. First is the allo antigens, so i.e. the antigens that vary between individual members of the same species, i.e. some of the HLA, okay, we've been talking about ABO blood type, Rh factors, and allo antibody is immunoglobulin that are that can attack these and alloantigens. Alright, so in terms of tissue transplant, we've been talking about blood transfusions forever, so we are not going to talk about it again. So the basic principles, I think by this time you guys should be quite adequately know what the principles behind blood transfusions. Okay, in terms of solid organ transplant, here is a graph that I pulled down from a government website. I think it's Transplant government. Here is just a FYI information. Really, you're not going to commit these type of knowledge into memory. Really, here is just a reference. So the majority of people are waiting for kidney. Okay, kidney is an organ that often fail. Okay, and then a lot of people are on dialysis and they are indeed waiting for a kidney. And the interesting part about kidney is that so you have any person would have two kidneys, okay, and the minimum functions that you need to maintain a good homeostasis is really about a quarter of a kidney. It's about 25%. If you have a 25% of functional kidney, that you can lead quite of a normal life. So that's why people can donate one of the kidney and they still have 50% of the kidney functions that can help maintain their balances. Okay, the second often common organ that need transplant is liver. Now liver here, we have a pretty big piece of liver, and again, this one can be a live donor, both kidney and liver. People with matching HLA type can donate a portion of their organ to another person to save that person. Okay, usually these have so those both the donor and the recipients can survive. But on the other hand, here we have heart and lung. Now those waiting lists are not as long, and really you are giving out the chances are you're giving out people that are diseased. Okay, no one can live without a heart, and no one can live without a lung. So someone must have passed away to be able to donate these organs. Like, there are different type of transplant type, and we've touched upon these concepts already earlier on. Here it's just a little bit recap. Autograft. Okay, autograft is basically transferring tissues between an area of the same individual, i.e. if you have a burn, okay, perhaps on a area that is more visible, usually the physicians can take out a piece of skin, okay, from perhaps an area that is more often being concealed, okay, with clothing, and then transplant to the damaged area where are more visible. So these are autograft. Okay, these are transplant. It's basically yourself. Okay, it shouldn't be any problem in terms of rejection. There's no way to reject your own. Not no way, but the chances are not very high. Right, isograft or syngraft basically is getting from a twin. Okay, basically with identical homozygous twin. So having a twin, it's good, not too bad. Okay, even though you growing up with a twin, you fight for everything. But you guys are basically the two of the siblings, twin siblings in this case, are a backup. Basically, it's a backup for each other. So the chances for survival, it's potentially a little bit greater. Okay. And then autograft basically is between genetically dissimilar individuals in the same species. Okay, so these would be recognized as non-self and termed non-histocompatible. Now, without any intervention, okay, without any immunosuppressant drug, these are not totally compatible in many cases. And xenograft is not done in human. Okay, we're not grafting anything from a different species into humans. Okay, the chances of rejections are even higher, so there's almost no way to graft that, at least in today's medical technology. Alright, so here is a recap of the MHC alleles. So basically we're talking about having parents giving birth to 4 individuals. Basically you have about 1 in a 4th chance that share both haplotype with a sibling. Okay, 1 in a 4 chance, and in about 50% chance you share 1, and then another quarter of a chance that you don't share anything at all. So that is the so here this one and this one are not sharing. Okay, so about 1/4 share, half share, but one of the haplotype, and then 1/4 of not sharing. So a similar concept that we mentioned in the last topic. Alright, so to find a matching HLA class 1 and class 2 is essential to improve outcomes of transplantations. Now, there are different ways to do the screening, i.e. the serotyping using antibody-based screening, mixed lymphocyte reactions, test lymphocyte activity, and genotyping. Okay, basically you are sequencing the entire gene using technology such as the PCR and DNA microarray. Now we'll talk about these technology once we are moving into the biotechnology topics, which is happening basically next week. Okay, next week we're going to start talking about biotechnologies. Right. So what are these screenings? Okay, HLA typing by serology. Basically, they are using antibodies, okay, treated with the cells, and the process is to see if there is a complement-mediated cytolysis, okay, when they are bound by HLA-specific antibodies, okay, so it's antibody-mediated complement systems, and if they are not compatible, basically, the cells will lyse, okay, and these cells will be treated with different fluorescent dyes, okay. Don't worry too much about ethidium bromide and all these dye. Basically, some of those are DNA staining compound, especially ethidium bromide. So it intercalates DNA double helix. So basically, ethidium bromide are intercalating in between the DNA pieces here. So when it fluoresces, basically there is a cell damage. The DNA are being leaked out. So that's the principles of HLA typing by serology. Now you can also mix, okay, lymphocytes. You're basically mixing peripheral blood mononuclear cells of a donor and the recipient, so basically mixing lymphocytes, okay, mixing it together, and then see if there is after a couple of days, you see if there is any type of changes in terms of proliferation, measuring T-cell proliferations. If the CD4 T-cell recognize the differences between the HLA class 2 differences, it will proliferate. Alright, and then also measure the T cell cytotoxicity. Alright, counting the T cells if that in response to HLA class 1 differences. Okay, so these are still the basic principles that we've been learning from the adaptive immune lecture. That's why that lecture was very, very important. Okay, everything basically is based upon those concepts. Okay, right. Now, moving on into different types of rejections, here is just a rundown of a list. We have hyperacute, acute, and chronic, and we're going to talk about each one of those in a little bit greater detail. So first of all is the hyperacute transplantations. In terms of the time frame, it's basically almost immediate or within a day. Okay, the reasons for these type of rejections, usually there is some type of a pre-existing antibody already, okay, so they will be against the basic ABO markers. So basically, if you have a unmatched blood type, okay, when you're doing transplant, the first thing is looking at blood type. If you have an unmatched blood type, okay, these ABO marker are also expressed on endothelial cells of blood vessels, and those blood vessels are usually carried over in a transplantations process. Okay, here's a kidney, there's the blood vessels here. Many times you are making connections, okay, mimicking connections between with the blood vessels, okay, to hook up the replacement there. Okay. Now, so blood type is the first things to match, and then now secondly, if and then there's also the HLA1 molecule. Okay, these are also expressed on endothelial cells. If those are can also be generating these immune attack response. Now, so what happened is that there is existing antibody that bind to these blood vessels throughout the graft. So you have antibody Y that going into and attacking the endothelial cells in the blood vessels, and as well as some of the tissues on the transplanted organ. So the immediate effect is leading to clotting, okay, and phagocytosis and hemorrhage. Okay, hemorrhage. You're basically cutting up or destroying the blood vessel that are supplying the blood to the new organ. So it is pretty a serious type of rejections. So why are there pre-existing anti-HLA antibodies? Okay, they can be arised from pregnancy, blood transfusions, or previous transplantations. So we're going to talk about that in detail in the next slide. Are there any treatment? No, because it is very serious one. Okay, and also happens very quick, and at that point you really just have to extract the organ. Otherwise, it would be quite lead to pretty bad situations. So what you can do to prevent these things happening, so must screen for patients against donor in order to avoid hyperacute. Cross-match test, okay, mixing, all right, the serums together, okay, looking at the reactivity between B cells and T cells, okay, does the recipient have antibodies that to react with donor, right? If looking at the complement fixations, okay, looking at if there's cell lysis, okay, lymphocyte lysis occurring. And we can also look at the panel-reactive antibody. Okay, we're looking at cross-matching the recipient and host serum with cells from basic representative panels of the individual. So looking at how much of these panel-reactive antibody are existing. And the higher up the percentage is, that means the patient have a antibody against more HLA types. So this type of a panel contains many different types of HLA. So really you're looking at that serum. Okay, so here for example we have 1, 2, 3, 4, and then you have the serum containing antibody. The more it can generate a type of a binding that indicate it can against more HLA, and that means it's more difficult to find a compatible donors. Okay, so the higher the number, the worse. Okay, the more difficult. Right, so talking about tracing back a little bit of why we have anti-HLA antibody. First cases is pregnancy. Okay, so pregnancy is a trauma. Really, that's very a lot of body changes at that time. Doesn't matter if you are if not you, if the mom is going under C-sections or natural birth, there's a lot of changes, a lot of injury is happening to the area, and in those cases the mother may be exposed to fetal cells and proteins, and since they contain some type of paternal HLA. Now, depending on how much of a differences, so it may act as a adult antigens and start elicit some type of a mild response. Those are pretty mild usually, and the mother usually won't feel anything, because even if there is some type of a crossing between the proteins, the amount is very small, and the mom can generate antibody and clear it up. But having it generated the antibody, going back through the basic concepts, there is memory. Okay, the next time seeing similar HLA, the problem will occur. And then the second one is transfusions. Okay, blood donors are matched for ABO, but not necessarily matching for HLA. So infusion of HLA incompatible leukocyte and platelet, along with the rest of the blood, may generate anti-HLA antibodies. Okay, so multiple transfusions, people that are have received a lot of transfusions in their lifetime, it tend to have generated antibody that against many different types of HLA. Okay, and therefore making them having a more difficult time to find a appropriate donor if they need one at the time. Okay, the third cases is prior transplantations. If you had transplantations before, okay, that also increases the percentage PRA, and that also makes it more difficult. Now imagine having matching one organ in your body that is difficult enough if you need to receive another one from a different person's. So the chances are, to carry 3 types of HLA, the chances of rejecting is much higher, okay, compared to people who don't have any prior transplant. Now we're going to the second type, which is the acute. So that one was hyperacute. This one is acute. Acute is caused by host effector Tcell, okay, responding to HLA differences between the donor and recipient. They're still kind of fast, okay, but it takes some days to develop, okay. Often what happens is begin with the destruction of endothelial cells lining the graft tissues, and then following by generating necrosis, okay. Necrosis is bad. Basically, it's a bad death, okay. During necrosis, there's a lot of cell components being released and can further generate inflammations and down the road. So it is not a desired outcome at all. So it takes a couple of days, okay, several days to develop. And the reactions, it's somewhat similar, delayed in a way that mimic some type of a type 4 hypersensitivity. We talked about it last week. So in this case, it's the whole CD8, okay, will be fighting against the donor's HLA1 and the host CD4, fighting the donor HLA2 class of the protein. Right, to prevent these things happen, you can start with inductive therapy, i.e. immunosuppressants. We'll talk about some of the basics of immunosuppressants toward the second half of this topic. And to treat, okay, we can also use an immunosuppressants and anti-T-cell antibodies. Now, what is the exact role of T-cell in acute rejections here? We kind of touch upon here. Let's look at this graph here. Now, this graph here, we have a kidney being grafted with dendritic cells being, residing in that kidney, and a little bit of the the blood vessels, okay, attached, and urethra being attached to the original organ, or the donor organ. Now, when these are being connected, the dendritic cells will start migrate out from the renal blood vessels and enter the recipient's circulations, and then ended up leading to the spleen, where they activate effector T cells. And it is at those spleen where the recipient T cells start seeing, okay, there is something different, okay, there's something different. So the donor HLA peptide is screened, against the T cells, for basically there is a reactivity, is being generated. Now, once that happened, the effector T cells will leave the spleen, and then coming out, and then home into the donor organ, and start doing its job, okay, basically initiating cell-mediated immune response against the transplanted tissues. So it will activate also Th1, okay, Th1 T cells will activate macrophages, causing inflammation, phagocytosis, and CD8 cytotoxic T cells would start actually killing the cells of the transplanted tissue. So you ended up losing that organ eventually, if you don't do anything, or any immunosuppressant therapy to it. Right, here is just again some FYI graph here showing you, hey, there is different type of rejection, showing it on a histologic histological type of a graph. The first one here, the lymphocytes, it's these little black or purple dot, depending on your screen and how you see it. These lymphocytes are going around the arterial, okay, so they are causing inflammations and of the arterioles. Middle panel, they're surrounding the renal tubules, okay. Again, there's a smooth muscle and that are being carried over during the transplantations. And the bottom panel, there are the T lymphocyte anti-CD8 antibodies. Okay, so here with T lymphocyte with this anti-CD8 antibody. So it basically all these little brown stuff here. Again, don't worry too much about this graph here, just for you to visualize a little bit. Now, and then the third type is chronic transplant rejections. Okay, it could happen from months to years following the treatments. Okay, this is usually again happening on the blood vessels, the graft arterial occlusions. So slowly there is a accumulations of atherosclerosis, or so-called graft atherosclerosis, thickening of the blood vessels. Now, really what happened is that these blood vessels again carry the donor HLA, and slowly they would begin to recruit inflammatory cells to the area, and causing slowly infiltrations of these macrophages will make the blood vessels to proliferate. Okay, basically it's thickening of the blood vessels, and start having productions of collagens by fibroblasts, and eventually leading to narrowing of the lumen of the blood vessel that is responsible for supplying blood and nutrients to the new organ, or to the donor organ. So slowly, it will lead to the organ being ineffective, being slowly leading the organ to death because of lack of proper nutrients supply, lack of proper blood supply. Okay. So there are different type of so-called allo recognition, recognizing the foreign or the donor type of HLA. First one is direct allo recognitions. Direct allo recognitions here shows in the graph if is your recipient CD4 CD8 T cell directly binding to donor cells. Okay, in many cases this is showing your the dendritic cells, the antigen presenting cell, so directly recognizing. All right, there are also indirect recognitions of the donors HLA complexes or peptides. Right, so in this case, showing you is this dying cells actually is the donor, DONOR, donor dendritic cell. So when they die, when they die by apoptosis, which is really a normal things to happen, they won't live forever. And when those things happen, they will start releasing these HLA types, and peptides, and these released HLA will be picked up by recipient dendritic cells, and then being processed through the vesicles, and then further presenting back on the donor recipients. Okay, basically it's almost like recognizing a bacteria type of a process. Okay, they're recognizing, eating it up, chewing it up, presenting back, and then you present it and being recognized by your CD4 T cells. Alright, so here is the follow-up. Okay, once these things are being presented to CD4 T cells, so you can either activate going through the macrophage branch, which is not shown in here, and then you can also go to the antibody production branch, where you have stimulations of productions of antibody that now can attack the HLA class 1 of the donor's type. So that is generations of antibody. So that's pretty straightforward. You either attacking it with the real T cell, not real T cell, with the cytotoxic T cells, or you are generating different antibodies, for those different HLA when you don't have proper immunosuppressant drug being administered. So this whole process, I think, is really mimicking, when you seize a bacteria, basically they are foreign, very similar process. Now, we're going to talk about very briefly on immunosuppressants. Okay, a lot of the drugs are not specific for as a immunosuppressants. Basically, corticosteroids. You learn it. You learn it again and again. So I'm not going to, talk about it in great detail again. So basically, it's just having anti-inflammatories. Okay. And then cytotoxic agents, okay, they mostly interfere with DNA replications. DNA replication, they kill proliferating lymphocytes, okay, kill T cells, basically. And then you can also inhibit T cell activations, okay, if they're not activating, so okay, they would not be so bad. And you could either use small molecule compounds, okay, or also use anti-T cells antibodies, okay, that's a little bit more fancy, but either way you're preventing its activations. So these are the basic principles, okay, basic principles of immunosuppressant drugs. So here is just one summary slide on the corticosteroid. So it inhibits NF-kappa B transcriptions and then lead to down regulations of many cytokines. Alright, cytotoxic drugs. Okay, I will talk about these drugs again separately in other in your GEM module again. So this week is really a companion week for my other material. So these are the rundown of these drugs: azathioprine, mycophenolate, cyclophosphamide, and methotrexate. So you've seen methotrexate somewhere, okay? These are just all cytotoxic drugs, okay? They are not specific, particularly for as a immunosuppressant. It can be used as an immunosuppressant, but it also used in many other places, particularly methotrexate. Basically, it's just kill cells, okay? They damage all tissues and body. They are not selective. Normally are cells that are in active cell divisions, i.e. things or cells that are in the bone marrow, intestines, and hair follicle, and leading to different types of unwanted side effects such as anemia, leukopenia, thrombocytopenia, GI effect, and hair loss. Alright, so pretty basic information here. So let's look at the inhibitors of T cell activations. So like I mentioned earlier on, these include some of the small molecule drugs such as cyclosporins and tacrolimus. Now you will see a very similar slide that I will present in the GEM module again. And in brief, okay, what is happening in this graph is that there is this protein called calcineurin. So what it does is to dephosphorylate this ANFAT, okay, ANFAT, and then ultimately leading to the activations of ANFAT binding elements on the DNA, which encode for different type of cytokine, particularly IL-12, okay, not IL-12, IL-2, and that would lead to T cell activations, VATION, and proliferations. And what these small molecule does is to forming complex with another neighbor here, cyclosporin will form a complex with cyclophilins, and tacrolimus will form a complex with this FKBB12, which is a some type of small peptide. Now once these drugs form a complex, it will inhibit bind to calcineurins, which prevented it to do its normal actions, therefore ultimately preventing NF activations, preventing IL-2 synthesis, and prevent T cell activations and proliferations and differentiations. Okay, so that is the pathway of how these small molecule drug works. Now we're gonna revisit that later on in other class, so don't worry too much in here. Then we have anti-T cell antibodies. So basically, you have an antibody that are against the T cells. And these include some of the polyclonal antibodies. Those are a little bit older type, and they contain antibodies that are against multiple T cell antigens, CD2, 3, 4, 8, 11. Many. You can bind to. Sorry, this is not the right one. So you have a T cells, and then you have the antigen binding. Okay, and so you have antibody binding it to a different type of CD marker there. And the side effects, okay, because they are not quite specific, can lead to a whole range of side effects, including leukopenia, thrombocytopenia, pruritus, itchy, even malignancies, okay, and serum sickness as well. Now we have better, okay, they are monoclonal antibodies, okay, all the maps there. So I don't want to make you memorize these names. Given the current circumstances, okay, we all know now that we are going to have online classes, okay, for a long time, okay, for the rest of the semester, basically. So I don't want you to memorize these names. The main message here is that they are binding to different CDs on the T cells, and also they can particularly they can also bind, okay, some of the anti-CD2, they can bind and antagonize IL-2 receptors that are on the T cell. So on the T cell, remember, T cells receive IL-2, okay, and then they lead to proliferation, differentiation, activations. Okay, if you block those receptors, you're basically stopping the activations process here. All right. So don't worry too much about the human marine part. These are just like FYI type of informations. Alright, coming to the last slide of this topic, we are going to look at two different types of tissue that are considered to have the so-called immunological privilege. Okay, the first one being is cornea. Now, cornea has some special immune surveillance that are suppressing inflammations. Right, the idea is that any type of inflammations that are happening on the cornea will lead to cloudiness on it and lead to impaired vision. So the eye has evolved an immunological environment that suppress inflammations while maintaining sufficient protections against pathogens, such as using having the lacrimal gland, secreting different the tear have lysozymes, and all these can help fight pathogens. So that is one of those mechanisms. And also inside of the eye we have aqueous humor that contain TGF beta. These will cause the dendritic cells in the area to downregulate the T cells stimulatory factors, okay, leading to less T cell activations. So the success rate, okay, for the cornea transplant, even without HLA matching, also immunosuppressant, is 90%. Okay, very, very high. So there's always some type of a advocacy, trying to persuade people to donate their cornea when they pass away, because 90% of the chance this cornea would be useful for someone that need to see clear with a new cornea. So that is one of those ideas. Another tissue here, we have liver. Okay, hepatocytes usually express very low level of HLA protein and no HLA class 2 proteins here. So really no HLA matchings you need to do. Basically, the first things you need to match would be the ABO blood type, okay, because again there are some blood vessels are being carried over to during the transplantations there. Okay, so now finding ABO blood type, it's much easier. That's why there are a lot of movies or TV shows are depicting some relatives, even perhaps sometimes husbands and wife, can sometimes donate liver to each other because the matching are not as you don't need as much of a good match to donate the liver. Alright, so that is the end of the rundown for transplantations, the concepts behind it, and how T cells and antibody mediate rejections. And let's go to look at some of the basic questions like usual, okay? Alright guys, we are continuing our millionaire game for this week, and we are going up to the $1,000 questions, okay? And let's see the questions. Hyperacute transplant rejection is directly caused by A. Antibodies, B. CD4 T cell, C. CD8 T cell, and lastly D. Dendritic cells. Okay, so this is pretty easy assessment to see if you're listening again for the past about 40 minutes or so. So what is hyperacute? So that is really quick, okay? Think in terms of what reactions is really quick, okay? It's T cells quick, okay? It's Alright, so give you 5 seconds. I think I've given you enough hint. 1, 2, 3, 4, 5, and the final answer is A, antibodies. Now, to be more precise, are pre-existing antibodies, okay, those that are already present in the recipient's blood, and those will be mounting a very, very quick attack against the donor tissues, alright, and leading to hyperacute within a day happens. Alright, let's move on to the next question, which is the $2,000 questions. Which mechanism is not utilized in immunosuppressant? Right, so this is a pretty basic assessment again. First, A, killing the T cells, B, blocking IL-2 receptors, C, inhibiting IL-2 productions, and D, block NF-kappa B receptor. Okay, and I'll give you a little bit time to think about it. So we are inclusively thinking about both biologics, i.e. antibodies that are against T cells, and we are also thinking about small molecules, okay, that can interfere with some T cell process. Alright, do you have a final answer yet? Okay, the final answer, I think you have it, is D, blocking NF-kappa B receptors. Now, so here is the catch. So NF-kappa B are being interfered, but not at the receptor level, but at the transcription level by different type of corticosteroids. Okay, so I hope you understand this very important key point. And without further talking, I am wrapping up for this week's lecture. Just to summarize what we did this week, we look at autoimmune diseases with 2-part video, and this one, the transplant, with 1-part video. And that is for all. And I'll see you next week a lot in GEM and in this class. Bye.